Dynamic DNA. Here with us today we have two scientists who will be talking about the relationship between DNA and genes and chromosomes. Please welcome
I have been working on a study about DNA and chromosomes and would like to share some of it with you.
Here's your model. Would you like to point some things out?
Yeah. DNA can be found in parts of the chromosomes. The relationship between them would be that DNA exists in chromatin. The chromatin folds up and make chromosomes, which are located in the nucleus of the cell.
Welcome back to today's show. Our next guest, also a scientist. Please welcome Dr. Moore. Explain to us the relationship between DNA and genes.
The relationship between DNA and genes is that DNA contains the genetic instructions that help develop and function living organisms and some viruses. Genes are DNA segments that carry the genetic information. Genes are the masses of DNA that direct certain things. A way to think of this is that genes are like the orders in the military and DNA is the words in the orders.
Tell us about a positive mutation.
A positive mutation would be a mutated gene that some individuals carry. They produce the instructions for a protein that prevents HIV from infecting the person.
Next, Dr. Bote will be talking about negative and neutral mutation.
A neutral mutation is one that has no effect on fitness. An example of a neutral mutation is having one green eye and one blue eye. It doesn't give any advantage or disadvantage. You can still survive with it. A negative mutation is which is harmful to the organism. The result of a negative mutation could cause errors in protein sequence, creating partially or completely non-functional proteins. Certain mutations can cause cells like germ cell and somatic cells to become malignant and thus cause cancer.
The last thing Dr. Bote will be talking about today is substitution.
Substitution is when one of the bases is substituted for another. For example, the pink is A and the green is G. I'll be taking out the pink and adding another green to substitute for the pink.
Now, Dr. Moore will give us an example of deletion.
Deletion is when one base is missing. So I'm going to take out the T.
There you go.
And there you go. Deletion.
Tell us about addition.
So addition is when one base is added. I'm going to add another G. And there you go. Addition.
Please welcome Dr. Bote to the stage. Thank you both for coming.
Our pleasure.
Well, that's our show for today. See you all tomorrow. Goodbye.